Pollen tube was discovered in Portulaca and it is first observed by G.B. Amacy. The only answer is G.B. Amacy, discoverer of pollen tube. Maheshwari, plant embryologist. Camerarius is discoverer of reproductive structure. Navashkin report double fertilization. Navashkin was associated with the double fertilization. Navashkin is concerned with double fertilization. Next one. Pollen tube actually develops over the stigma. This is the stigma and this is pollen grain. And pollen grain forms pollen tube. Pollen grain forms pollen tube. And this is ovary. And within ovary, ovule is present. This is ovule. And within the ovule, embryo sac is present. Inside the ovule, embryo sac is present. This is embryo sac. In embryo sac, this is egg cell. This is synergid. These two are the two polar nuclei and three antipodal cells are there. And in synergid, you see a finger-like structure. A structure present with the finger-like projections. This is known as filiform apparatus. This is called filiform apparatus. In which cell? That is synergids. The chemicals of this structure, it releases chemicals, and these are calcium boron inositol complex. They stimulate the growth of pollen tube. They are responsible for the growth of pollen tube. That's why the growth of pollen tube is chemotropism. Pollen tube show chemical induced movement of growth. Pollen tube shows chemical induced movement of growth. What is shown by the pollen tube? Chemical induced movement of growth, and those chemicals are calcium boron inositol complex, and chemotropically active substances are secreted by the filiform apparatus of synergid. Filiform apparatus of synergid. Germination of pollen grain on stigma. Stigma is a living tissue. Stigma is living tissue, and any structure which develops on the living structure on living tissue, that is known as in vivo germination. That is known as in vivo germination. So development or germination of pollen grain, germination on pollen grain on the stigmatic tissue is in vivo. In situ, that is inside the structure. In vitro, that is within test tube. In vitro, that is within test tube. On living structure, that is in vivo. Pollinia. Pollinia are found in Calotropis plant. Pollinia are found in Calotropis plant, and these pollinia are actually sac-like structure. See this structure? This is disc-like structure, sticky disc-like structure, and from this stalk-like structures are formed. It forms a stalk-like structure, and two sac-like structures are there. This structure is known as pollinia. These are the pollinia in which pollen grains are present. In this structure, number of pollen grains are present. This disc-like structure is corpusculum. This disc-like structure is corpusculum. And these stalks are known as caudicle. These are called caudicle. and these sac-like structures pollinia. These are pollinia. Sac-like structures are called pollinia. And this structure is carried as a whole by the insect, and the entire structure is known as translator apparatus. This is known as translator apparatus. And the mechanism of its transfer is known as translator mechanism. The mechanism is known as translator mechanism. 20th question, that already described with the previous one. That is translator mechanism is seen in Calotropis. It is seen in Calotropis only. Male gametophyte of angiosperm shaded, that is pollinate at. The correct answer, in 2 or 3 celled stage. In 60% angiosperm, it is in 2 celled stage. In 40% angiosperm, in 3 celled stage. If it is released in 2 celled stage, then you see this is vegetative cell, and within the cell a floating structure is generative cell. Generative cell. And first one is vegetative cell. If it is released in 3 celled stage, then this is vegetative cell and 2 male gametes. 2 male gametes are there. They are formed from the generative cell. This is 3 celled stage and these 2 are the male gametes now. So count number 1, number 2, number 3, that is 3 celled stage. 2 male gamete in vegetative cell. The vegetative cell is 1 cell, 2 male gametes are 2 cell, that is 3 celled stage. But 60% plants of angiosperm pollinate their pollen grain in 2 celled stage. 2 celled stage. Question number 22. In an angiosperm, how many microspore mother cells are required to produce 100 pollen grains, but it is in family Cyperaceae? This is applicable. This is the different case. Actually, 1 PMC, that is pollen mother cell, 1 pollen mother cell usually produce 4 pollen grains or microspore by which process? Meiosis. But in Cyperaceae family, 1 PMC produces only 1 pollen grain. Only 1 pollen grain. This is the normal case. This is the normal case, but this is exceptional case. This is exceptional case. Family Cyperaceae, exceptional case. That is, its PMC produces only 1 viable pollen grain. So straightway, that is to produce 100 pollen grain in Cyperaceae, 100 pollen mother cells are required. That's why the number of meiosis is as the number of pollen grain. In that case, how many microspore mother cells are required? Answer is 100. Whereas in other normal cases, 25 is sufficient. So for family Cyperaceae, answer is 100, and other normal cases answer will be 25. Ategmic ovule, ovule without integument, that is ovule without integument, that is ategmic ovule, found in Loranthus and Centellum album. That's why answer is 4. Loranthus and Centellum album, they have ategmic ovule. Most common type of ovule is bitegmic, that is covered by 2 integument. This is most common. Bitegmic ovule is most common in angiosperm. This is most common type of ovule. Most common ovule is bitegmic. Now come to the next question. Hilum, chalaza, micropyle lie on same longitudinal axis. See this one. Actually this structure is concerned with the type of ovule. This is the ovule nucellus. This is ovule nucellus. This is integument 1 and 2. This is the integument 1 and 2. Sorry. This is integument 1 and 2. See this structure actually. You see this is the body of ovule. This is the body of ovule. This is funicle. Funicle or ovular stalk. This is funicle or ovular stalk. Then this portion is attachment of funicle with the body of ovule known as hilum. This is known as hilum. Then a small opening that is known as micropyle. This small opening is known as micropyle. And opposite to micropyle, chalaza is present. Opposite to micropyle, chalaza is present. The body of ovule is placed straight over funicle. So it is orthotropous ovule. First of all, this ovule is orthotropous. This ovule is orthotropous ovule. Second, see this one. That is hilum, chalaza and micropyle. They are on same longitudinal axis. Yes, they are on same longitudinal axis. This is hilum region. This is chalazal region. This is micropyle. Hilum, chalaza and micropyle, they are on same longitudinal axis. That's why answer is orthotropous ovule. What is the direction of micropyle in anatropous? Anatropous ovule is most common type of ovule. Anatropous ovule is most common type of ovule. This is the nucellus of ovule. It is covered by integument 1 and 2. And the body of ovule is bent over funicle. See this. This is anatropous ovule. This is anatropous ovule. The body of ovule is bent over the funicle. This is funicle and this is the ovular body. The body of ovule is bent over the funicle and this is the micropyle. See this one. It is not upward, it is downward. It is downward. So answer is 2 because body of ovule is bent over the funicle. Bent over the funicle in this manner. In orthotropous it is straight. This is body of ovule, this is funicle. The orthotropous ovule, the body is straight, whereas in anatropous ovule it is placed inverted. So the direction of micropyle is downward. Then ovule, let's see this question. The ovule becomes curved. So nucellus, embryo sac lie at the right angle to the funicle. Right angle. See this word. Right angle to the funicle. This word is important. That is right angle. This is body of ovule, slightly curved. This is integument 1, integument 2, integument 1 and 2. Now see this structure. The body of ovule is placed transversely. That is right angle to the funicle. This is the right angle to the funicle. This answer for this question is hemianatropous. Hemianatropous. Right? In hemianatropous, that is semi-placed. Anatropous, this one. Orthotropous, this one. And if it is completely curved, then amphitropous. Which of the following represent the ovule where the embryo sac becomes horseshoe and funicle micropyle close together? See this one. This is the body of ovule. It is curved. It is curved, right? And the prominent portion of any ovule is nucellus. And inside the nucellus, embryo sac or female gametophyte is present. And it is also curved. This female gametophyte is also curved. This is ovule. It is curved. An embryo sac inside the ovule, it is also curved. This is embryo sac. This is embryo sac or female gametophyte. Female gametophyte, and both are curved. Both are curved. So this is Amphitropus. This is Amphitropus ovule. Which one of the following is most common amongst? I have already mentioned that is Anatropus is most common. Anatropus ovule is most common amongst angiosperm. It is found in 82% plants. This type of ovule is found in 82%. It is like this. This is Anatropus ovule, most common ovule, and it is present inside the ovary. It is present inside the ovary. Cercenotropus ovule is found in cactus, known for long funicle, long and coiled funicle. Atropus is found in Polygonum. Atropus is found in Polygonum. Amphitropus in Alisma. The Anatropus ovule is most common amongst angiosperm. In the ovule, there is only one single archisporial cell. Nothing to be discussed. It is single celled. It is single celled and hypodermal. This is epiderm of nucellus. This is epiderm of nucellus. And these are the integuments. This is archisporial cell. Now next one, point out odd. Nucellus, ovular part, diploid. Embryo sac, haploid, female gametophyte. Micropyle, part of nucellus. It is part of nucellus. Pollen grain, they are from stamen. They are from the stamen. These three structures are concerned with the ovule, whereas this is concerned with the stamen. And the question says which one is odd. So definitely pollen grains are the odd structure here. Pollen grains are odd structure here. They are the part of male gametophyte. Pollen grain represents male gametophyte, whereas these three structures are concerned with the ovule. They are concerned with the ovule. Embryo sac occurs in, answer straightway, ovule. This is nucellus of ovule. This is nucellus of ovule, and within this nucellus, embryo sac develops. Embryo sac develops from megaspore mother cell. Embryo sac develops from megaspore mother cell. This is embryo sac. Location of embryo sac or female gametophyte towards micropylar end. This embryo sac is 7-celled 8-nucleated structure. 7-celled 8-nucleated structure. This is haploid structure. It is inside the diploid structure. This is embryo sac or female gametophyte. Embryo sac is also called female gametophyte. This embryo sac is also known as female gametophyte. Now, 8-nucleate embryo sac. Yes, It may be tetra, it may be mono, it may be bisporic. So that's why answer is 4. That's why answer is 4. If develops from single megaspore, then monosporic. If develops from a cell with 2 megaspore nuclei, then bisporic. If develops from a cell which has 4 megaspore nuclei, then tetrasporic. And all possibilities are there. They are not needed for the need purpose. Female gametophyte. Female gametophyte is represented with this embryo sac. Female gametophyte is known as embryo sac. This is ovule, nucellus. These are the integument. These are the integument. This is the micropylar region. This is micropylar region. And inside the nucellus towards micropylar end. Inside the nucellus towards micropylar end, this is female gametophyte or embryo sac, comprises of 7 cells, that is cell number 1, this is egg cell, this is synergid, and this is the largest cell in which two nuclei are there. This is the largest cell, 4th cell, that is known as central cell. This is central cell with 2 nuclei, and these are the antipodal cells, 3 antipodal cells. So count it, 1, 2, 3, 4, 5, 6, and this is the 7th cell in which all these cells are present. This 7-celled 8-nucleated structure, 7-celled 8-nucleated structure is embryo sac. 7-celled 8-nucleated structure. 8 nucleus, 2 nuclei are in polar cell actually. 2 nuclei are there in central cell. Nucleus 1 and nucleus 2. 2 nuclei are found in central cell. Then each cell with 1 nucleus, each cell with 1 nucleus. So answer is embryo sac. Arrangement of normal embryo, arrangement of nuclei. Arrangement of nuclei, see this structure. This is egg cell with 1 nucleus, synergid 1 nucleus, then this is 1 nucleus of synergid, then 2 polar nuclei are there and 3 antipodal cells with 3 nuclei. So check it. Towards micropylar end, 3 cell with 3 nucleus. This is the largest cell in center of this cell, 2 nuclei are there, and these are 3 antipodal cells with 3 nuclei. So this arrangement is termed as 3 plus 2 plus 3. Answer is first one. The first one is the correct answer. Through which cell of embryo sac does the pollen tube enter? Yes, check it. This is the stigma, style, and the swollen base is ovary. And this is the ovule. This is the ovule. And this portion is micropyle. And within ovule, embryo sac is present. Within ovule, embryo sac is present. Embryo sac is haploid structure. This embryo sac is haploid structure inside the diploid ovule, and this is egg cell, synergid. Now, pollen grain first of all reach to the stigma by the process of pollination. Then it germinates and form pollen tube. This pollen tube grows. Its growth is chemotropism, and it enters through micropyle. and through one of the synergid, and through one of the synergid, and through one of the synergid. So if we enlarge female gametophyte, this is female gametophyte egg cell, and this is the synergid. This is synergid, this is synergid. Synergids are known for filiform apparatus, and pollen tube enters. Pollen tube enters in ovule and through integument. Pollen tube enters through one of the synergid. This is the pollen tube and it enters through one of the synergid. That's why the answer is now quite clear, that is degenerated synergid. Persistent synergid means that survives even after the entry of pollen tube, but it is degenerated because through one of the synergid. The statement given below regarding true for female gametophyte, it is 8 nucleated 7 celled, right. Free nuclear division during development, right. It is situated inside the integument but outside the nucellus, wrong. It is present inside the nucellus. It has egg apparatus situated towards chalazal end. This is wrong. It is towards micropylar end. It is present towards micropylar end. So these two statements are wrong. Answer is we have to select the true only. We have to select the true options. In that case, answer is 1 and 2. This is the correct answer for this question. Then egg apparatus. Its location is micropylar end and it is 3 celled. Egg apparatus location is towards micropylar end. It is 3 celled, that is 2 synergids and 1 egg cell. 3 celled, why? Composed of 2 synergid cells and 1 egg cell. 1 egg cell or female gamete. These are the part of egg apparatus. Then starting from innermost part, correct sequence of an ovule. Starting from inner part, this is nucellus. This is nucellus. Just a minute. This is nucellus. These two are the integument. These two are the integument. Now this structure is ovule, and within ovule embryo sac is present. Within ovule, this is embryo sac. In embryo sac, this is egg cell. This is egg cell. These are synergids. So let's come. This is the innermost portion is embryo sac. So come egg, embryo sac. Egg cell is found in the embryo sac. Egg cell is present in embryo sac. So first is egg cell followed by embryo sac. And the embryo sac is covered by the nucellus. Embryo sac is present inside the nucellus. This is the nucellus, and nucellus is covered by the integument. So the correct sequence 2. Egg cell, embryo sac, nucellus, integument. Egg cell, nucellus, first egg cell, then nucellus, embryo sac. This is egg cell, this is egg cell, this is egg cell, and this is embryo sac. This structure is nucellus, and these coverings are integument. Our integuments are outermost layer. So answer is 2. Pollen tube. Growth of pollen tube, chemotropism. Chemotropism is shown by the pollen tube. For fertilization, pollen tube. Entry of pollen tube. Pollen tube normally enters through micropyle. This is most common, but it can enter through chalaza as well as integument. But the most common entry is from micropyle. And if it enters through micropyle, the process is called porogamy. If for fertilization, pollen tube enters through micropyle, the process is known as porogamy. What is known as? Process is termed as porogamy. Now, mesogamy. What is mesogamy? This is ovule. This is ovule. These are the integuments. If pollen tube enters through integument, this is what? Pollen tube, and it is entering in the ovule through integument. The process is known as mesogamy, and it is seen in cucurbids. If it enters through chalaza, then process is called chalazogamy. Chalazogamy is common in casuarina. Chalazogamy, that is pollen tube enters through chalaza. Double fertilization. This process was observed first in Fritillaria and Lilium. What is double fertilization? This is embryo sac, this one is egg cell, and these two are the polar nuclei. And this is egg cell. Other cells are not required. And pollen tube contains 2 male gametes. Pollen tube has 2 male gametes. This is pollen tube. It has 2 male gametes. That is male gamete number 1. This is male gamete number 2. One male gamete fuses with the egg cell. That is, male gamete fuses with the female gamete. This is one fertilization known as syngamy. This is called syngamy. Whereas other male gamete fuses with these 2 polar nuclei. This is known as triple fusion. Process is known as double fertilization. The process is called double fertilization. Now, number of nuclei participates in the endosperm formation. Embryo sac, and these two are the polar nuclei. This is egg cell. These are the synergids, and above 3 are antipodals. And nucleus number 1, this is nucleus number 2, and one male gamete of pollen tube. This is one male gamete of pollen tube with one nucleus fuses with these 2 nuclei. So how many nuclei are fused? One from male gamete, one nucleus of male gamete, and 2 nuclei of central cell, polar nuclei. Total number of nuclei participated 3. Then only the primary endosperm nucleus or pen is formed, which further develop to form endosperm. So here the answer is 3. Regarding the formation of endosperm. Generative fertilization, that is syngamy. It is called syngamy only. That is fusion of male and female gamete. Fusion of male and female gamete is known as generative fertilization, results in the formation of 2N zygote. Triple fusion is known as vegetative fertilization. It is called vegetative fertilization. It is called vegetative fertilization. And the last one, oily endosperm, oil containing endosperm. It is found in Ricinus communis, scientific name castor seed. And one more, that is Coccus nucifera coconut. After maturity it becomes oily. So there are two examples of oily endosperm, that is Coccus nucifera and Ricinus. That's why my answer is more than one. My answer is more than one, because Coccus nucifera is for the coconut and Ricinus communis for the castor. Both contain oil at maturity. So oily endosperm is found in them. Thank you. Thank you. Thank you.